So today we're going to talk about your ANDA or ours. So there's a lot of different people that may be out in this audience today. Maybe you are part of an innovation team within a branded company. Maybe you're a part of a sourcing group within a larger company that needs to find a CMO partner, or maybe you just want to come up with a brand extension. How do you do that? You can either develop your own within your existing R&D architecture, or you can partner up with a CDMO. If you want to transfer your product in, perhaps it's being commercialized somewhere else, and you were looking for another CMO partner, then you would transfer into us as well. We're going to talk about some of the challenges and some of the hybrid solutions that are starting to emerge that we're starting to see in the industry, and to give you some good insight as to how you can more seamlessly get your product onto the market faster. So let's start with the decision tree. So this is going to be kind of the decision tree that we're going to map off for the entirety of this discussion. First of all, and we're not going to necessarily limit this down to ANDAs. This could also be ANDAs as well as OTC products. So either one this kind of applies to, because we transfer both RX, ANDA products as well as OTCs into our facility, and we also develop all three of those. So just because you're seeing ANDA here, it could mean any of the above. So this is kind of the roadmap, or kind of the decision tree that we look at. And one of the first thing is, do you have your own ANDA? Do you have one, or are you just looking for a CMO partner? If you do, and it's currently commercially manufactured, then it's going to be a direct transfer into our facility. It's going to be pretty seamless. Now, there will be some complexities with that, depending upon whether it's an immediate release type product or a modified release product. We'll talk a little bit more about that in a minute. If you don't have it already manufactured and it's in the process of being reviewed by the FDA, or in the case of an OTC, it's about to come out of your R&D, ready to be commercialized, then there's going to be a different option where we're going to start to look at scaling up, and we're going to commercialize that, validate it, and then commercialize that. That's also something that we do as well. However, on the other side of the ledger, if you don't already have an approved ANDA and you're someone who's looking to expand your brand portfolio and you're looking for options to put into that, there's several different ways you can go about that. You can develop your own OTC within your own R&D organization, or you can partner up with a CDMO such as ourselves, and we can develop it for you. The other option is if it is an ANDA or a generic product that is regulated, that's on the OTC market, there might be the opportunity that we have that ANDA within our network already approved, because at my company we have about, I think there's around 72 different ANDAs that are already approved, and almost all of those are in the OTC space. So there's a good chance that we might already have that approved somewhere, and then it's simply a matter of commercializing it, and it's quicker to market. No need for the development, no need for the transfer. It's just a simple line extension for whatever company you're working with. So let's talk about these in a little more detail, the regulatory concept. OTC monographs. So the OTC monographs, the example of that would be your Tylenol, your 500 milligram APAP. Those are products that the FDA has generally considered as, they call it GRAS status, generally recognized as safe and effective. So those do not need to go through any sort of dossier or regulatory approval, submission and approval process, and certainly no biostudies are necessary. Another example of that would be your 81 milligram aspirin, these types of things. The next one, I'm going to skip down to the NDA. So the NDA is a new drug application. So these are typically done by a branded company where they take the drug from start to finish, and they do all the clinical studies to go ahead and get all of their label claims, and to show that their drug is safe and effective for the particular indication that they're applying for. Then there's the ANDA, that's the abbreviated NDA. That's essentially a generic product, a copy of the NDA product. And that's where our company lives in, is that particular product. Why does this matter? It matters a great deal when we start to talk about some of the things in the next couple of slides in terms of how you go about bringing your product into the market, because we're talking about timing and we're talking about cost. And we're going to show you some examples of things that we've actually done, products that we've actually worked on with other companies in order to bring these things into the market in a much faster method. One of the things that I can tell you as you walk around is that a lot of companies really are not aware that this is taking place. We've always just assumed that a branded company or another company is another company. They operate within their own silo, that they don't work with external partners, except for on a CDMO side or a CMO side, just to manufacture their product. But there's other ways to get to those products, because sometimes your CDMO already has those products. So we'll look at some of those examples as well. Regulatory pathway. So here's a basic idea. This is very basic on what you're looking at when you're talking about that. On a monographed OTC, if you transfer a product in, if you've got a monographed OTC being manufactured somewhere else and you're looking for it to be manufactured at our site, there is no regulatory dossier that needs to be submitted, so therefore you're going direct. We'll describe the steps of that in a little more detail in the next slide or two. Then you get into the immediate release and the modified. These govern the NDA and the ANDA pathway. So the FDA runs under their guidances, their SUPAC guidances for transfer of a modified release or immediate release. Immediate release product is one that has no sort of technology built into it in order to modify its drug release characteristics. It's an immediate release product like a Tylenol, for instance, acetaminophen or a paracetamol. That generally will be a CBE 30 on your transfer, because the FDA doesn't view that as a high-risk category for transferring your product. However, if you're in the modified or extended release category, that's going to be something that the FDA is going to take a much harder look at, and the guidances state that almost every case you're looking at a new biostudy. So if you do have an ANDA or an NDA or Rx, whatever, that is going to need a new biostudy and a PAS approval. So a CBE 30 on the immediate release is 30 days. The FDA has 30 days in which to come back to you and say, yes, you're approved, or no, we would prefer to take a longer look at this and move it to a PAS, or in most cases, it's usually you're on the market in 30 days for an immediate release. If you do have to go the PAS route, that's running about 9 to 10 months now, depending upon the classification that the FDA gives it under GDUFA 3, so it's about 9 to 10 months to get that through, and the critical part is you're talking about a biostudy. So that, you're talking not only the cost, but the timing associated with getting something like that done. So let's look at 3 strategic approaches that might exist. We can use your ANDA or your NDA and transfer it into our facility. And again, if it's an immediate release product, it's very simple to do. We bring it in, we run the gamut of batches in order to get stability and prove it, file it with the FDA as a CBE 30. And in almost every case, we're good to go within 30 days. However, if it falls in the modified release category, it's much more complex and costly. You can use our ANDA, so that's another thing that's emerging, is that some of the branded companies, even though they've had their brand on the market forever, they look in our portfolio and say, wait a second, you already have this approved modified release ANDA that's being produced in your facility. We can simply just have you manufacture it under our brand name, and that becomes now the brand. So it's a quicker way to get into the market, a much easier way to get into the market. Then we have hybrid solutions. So there's all kinds of different things. We have one product right now that it was a branded product that had been on the market for a very long time. The brand company decided that they wanted to change the process because it was much too archaic and costly. And they identified that it was going to take them biostudies, it was going to take them a couple of years in order to change their process. So instead, they licensed our generic process and product to transfer into their facility so that they could use that product and process in order to manufacture the brand at a cheaper cost. So that's an example of a hybrid solution. And it's something that outside the box thinking is something that we're seeing really gain traction within this space in the past few years. What makes the ANDA complex? So if we do have to go the ANDA route, we want to either develop our own. This is what happens. You have to match the reference product. So you have the NDA, you have the branded product, and that's what we refer to as the reference listed drug, and you have to match that product. We'll talk in a minute about the bio studies. There's scale-up challenges that go with that, and of course now if you have to develop your own, you're talking about speed to market. And a lot of people, especially at this conference, are looking for speed to market. They may not be looking at long-term, 2 to 3 year projects. They want something on the market for brand extension, line extension now. So this is an example, for instance, and we're going to show a couple instances of drug release, because when you start talking about especially modified release products, this is really the key driving factor, is the drug release, the dissolution. Is you have to match the drug release of the reference listed drug, because that ultimately is what is going to correlate to your in vivo plasma profile. And we'll talk about the biostudy here in a minute. This is another example of some of the dissolution apparatus, the drug release apparatus that we utilize in order to sort of mimic it in multiple pHs to give ourselves a better indication that we have matched drug release between the reference listed drug and the test product going into the biostudy. You want to make sure that all of that stuff is worked out ahead of your biostudy, because that is time and expense if you don't. So what is a BE? Bioequivalence study. So in order to make a generic product, the FDA has said, okay, the brand company has done all of these bio studies in order to prove blood levels, prove efficacy within their label claims. If you want to put a generic product out there, you're going to need to match it in a bio study. So what essentially happens is, in a basic two-way crossover study, is that you'll have, we're going to make up a number, 24 patients go to the clinic. They're going to go to the clinic twice. The first time they go there, they're going to take the brand product, and then the second time they go there, they're going to take the generic product. And what they do is they match the blood levels between the two within the same person, taking both tablets or both liquids, whatever the case may be, and they look for blood levels. They also require you to do it in two different scenarios, a fed and a fasted state. So, pretty self-explanatory. A fasted state is where they have an overnight fast before they dose the product, and that means your stomach's empty. And what happens is the pH of the stomach in a fasted state is around 1.2. It's highly acidic, and products will essentially flush through the stomach at every 10 to 15 to 20 minutes and get into the upper GI, which raises the pH to around 6.0. So that's a much different pH profile than in a fed state, where you now have a residence time of around 2 hours or so at a pH of about 4.5. So those are things that will absolutely affect the release and the absorption of the drug. So they want to see how the test product reacts to the different environments, compared to the RLD. Let's talk about the BE metrics real quick. They're looking at three different things. They're looking at the Cmax and two AUCs. We'll classify them into two just for argument's sake in this talk. The Cmax, that's the maximum concentration. So what they're doing is they're taking blood levels over time, over a couple of hours, and they're making sure that the blood levels never reach a maximum concentration more than the brand product is. You want to stay within a certain range of that, so you don't exceed it, and that has to do with safety and efficacy. The next thing is the AUC. That is the total amount of drug that ends up in the bloodstream, area under the curve, statistical measurement, and that's how they tell whether or not you have a bioequivalent product. Whether you've got a Cmax and your total AUC has to be within a certain statistical range to the innovator product. If the BE fails, you either have to reformulate it, or you have to redesign the study, or increase the population. And those last two things have to do with the statistics. So it turns out it's not as simple as everybody thinks. Everybody absorbs, everybody reacts to drugs in a totally different manner, even within the same patient. You may take one drug one day, you'll get a certain blood level, the next day you'll get a totally different level. Those are statistical variations, and sometimes you have to redesign the study, possibly even going, in one case, we had to actually do a fully replicated study where the patient comes in and they come 4 times instead of 2 times. They take the reference product twice and the test product twice. And then you measure those against each other to get a statistical average so that you can measure against the innovator. So the tech transfer part of this is where it gets real. So if we're not going to do the ANDA, we're going to go with a full tech transfer. What are we looking at from a basic standpoint? Regulatory alignment is the first thing you have to do. You have to define what regulatory strategy. We're going to look at that in the next slide, so I'm going to hold off on that. So once you get the regulatory alignment, then we would do what we call engineering batches. Lots of people call them different things. Demo batches, experimental batches, so that you can, at full scale, so that you can identify whether or not you're getting the same performance at your sending site that you are at your receiving site. The next thing is you're going to run stability batches. This is your pivotal stability. So before you launch any product or you go into your validation studies, you're going to want to make sure that you have stability, whether it's an OTC or whether you're going to file this as an ANDA under the CBE 30 or a PS rules. You're going to need to see stability of batches. How many batches you run is really dependent upon the product and the risk analysis, and a lot of times even the company's tolerance for risk, right? So some companies will want to see 3 batches, some are fine with just 1. It really depends upon what the FDA talks about the body of work or the stability, historical stability of the product. If it is a little shaky, you might want to run 3 batches. You might want to look at different raw material lot numbers within that and vary those. So we've done everything out to 6 batches sometimes to make sure that it's the same at the sending site as it is at the receiving site. And then once you get your stability, and usually 3 months is the marker, once you get that, then you've got lock and you're ready to roll into your validation, which is essentially pre-commercial. So the product transfer. So here's where the regulatory strategy does get a little murky. First of all, we talked about the differences between the immediate release and the modified release. So if it's immediate release, you're going CBE 30. If it's modified release, it's going to be a PAS. There's also a second level, and this really speaks to the number of batches that are required. The FDA made a shift in 2013, June of 2013. So if you have an ANDA that was filed prior to June of 2013, you were only required to produce 1 batch and get 3 months of accelerated stability in order to file that batch. After June of 2013, everything shifted. We went with the European model. It now became 3 batches in 6 months of accelerated stability. So that is it's a huge win if you've got a pre-2013 ANDA that you want to transfer, because now you only have to, under regulatory requirements, produce 1 batch and get 3 months of accelerated stability in order to file that CBE 30. And a lot of our ANDAs that we have within our portfolio are pre-2013. So let's talk about some real life challenges that we actually did. So we actually, we acquired an ANDA on the OTC market. It was actually an RX to OTC switch, so we acquired it many years ago. We brought it into our facility, and to show you how old it is, any of you who are process guys in the room, the blending step was actually done in a high shear granulator. It was a blending step, not a high shear granulation. They were so scared of blend uniformity that they did it in a high shear granulator. So when we transferred it to our facility, one of the things that you look at is the SUPAC guidance, and it has to be like for like, right down the line. Method of operation of each piece of equipment. So we took out the high shear granulator because it was a catastrophe in terms of product flow and some of your metrics of getting the product out the door. So we moved it into a bin blending system with an intensifier bar, and therefore it functioned the same way in terms of sort of getting some agitation going within the bin. Filed with the FDA, that normally would have been a PAS and maybe even triggered a bio-study. Probably not on this product, but it could have. But we claimed that this was a like-for-like change, and that we had, and we showed it throughout our CU, because we did the 40x3, the ISP guidance for that, and we did it with 3 batches. We could have only gotten away with 1, but instead we did 3 batches full scale for this, and it got through as a CBE 30. So that's one example of how we did it with an immediate release. And this is our bin blending system that we have at our facility. This is actually running the product. So modified release challenges. So this one was very interesting. So this is a PK profile, in case any of you have not seen that, just for visualization. This is a plasma profile of the test versus the reference, and you could see how the lines kind of match up to each other. Our product had a little bit more Cmax on the open. This is a delayed release product, just so we're aware. So this one was extremely challenging. This is actually a MUP system, so it is a pelletized system that's compressed into a tablet. We acquired the ANDA before it was ever approved, and there was no plans to ever have it approved. We got it through the finish line, got it through the FDA, and got it approved. But then we needed to figure out how to transfer it out of the original site, which required a bio-study. And we knew that going in, so we were prepared for the pain and suffering, which we incurred pain and suffering. So this is one example that we actually had to make the pellets at a third party, bring them in and compress them into a tablet and run the bio study. And it's very successful. So this is another example of how you can bring in another ANDA, but you might have to run the bio study, but you can be creative in using third party to manufacture certain parts of the finished product. So we're going to end up with our decision tree here. Before we go into a question and answer, we're swinging right back around. Again, this is really to sort of stimulate the innovative juices this talk has been about how you can bring your product to market. And there may be a variety of people in the audience who, we want to encourage you, don't think in old terms that, hey, we have our own R&D, we do this. You may be able to just find something that we may have within our portfolio that can get you there much, much faster. We do it every day. There's a lot of people that come to us and they say, I need this now, and they pick up the phone, they call us, and we have something for them, and whether it be on the OTC or the ANDA side, they throw their brand label on it, and we're ready to go. So again, that's the key decision maker right there, and it's on the left side. If you're not really interested in the control of it, you can just use ours. And even in cases where the product may have, say it's it's an ANDA liquid, you may have your own, you might want your own flavor. We can do that as well. We file that as a PAS, you can change flavors, you can change all that stuff. It becomes your own flavor. And that's the R&D group. So I think we put that in there to show that we actually do have an R&D group. It is an R&D group that we have. And we're based in Miami. Questions? Questions?
Thank you. It was really fascinating. You mentioned you mostly do this with OTC products, or that those are the primary base. Could you talk a little bit about whether you've looked at it for, GX, if you have much of that, or what some of the barriers are to having a bigger portfolio in GX?
Yeah, so actually the most we do is in generic pharma. The most of these are because of the ANDA, because that's where it becomes complicated. OTCs, of course, we do sell a lot of those into the market on the private label side, but those aren't complicated in terms of, everybody can develop those, and most of them are pretty simple. It's no secret. There's no special sauce in a 500 milligram APAP. But I think what I'm really trying to impress upon people today is that we do have a library of over 70 ANDAs, and we do this. And a lot of times they do become brands. And it's because of this complexity. It's just easier. It's easier. If a branded company or a company sees something that they are not currently involved in right now, they have a specific brand that they want to do some brand extension and bring a product into it that some other company has, they'll look to us and say, do you have this generic? And we could put our brand on that. And so that's exactly how this is playing out. So it's actually fascinating how many of the generics are becoming the brands. We even had one case where over COVID, there was a branded company. We were the largest generic private label supplier in the market. They had a problem at their API site overseas, and it shut them down. They could not get out of that. It was going to take them 2 years to get back on the market. So we literally, under our ANDA, produced the brand for 2.5 years until they could get back on the market. So they just said, look, we just need you to use our tooling and use your formula, and until we can get back on the market to solve this regulatory problem. And they did.